Here we go. So good morning, grade 12 students. In this session, we're going to continue with the exercises of the worksheet, the revision worksheet for the genetic chapter. So we finished the first three exercises in the last sessions, and then we're going to continue now and today with this exercise, question number 4. Question number 4 is talking about hemophilia B. Hemophilia B is characterized by the absence of the blood clotting, which may lead to significant hemorrhage. It's linked to the disease to the absence of a clotting factor, which is called factor 9, whose density is controlled by a gene located on the non-homologous segment of the X chromosome. Honhu allele is found in the non-homologous segment of the X chromosome, so no need to justify it unless they ask for it. This abnormality is found, or this gene is controlled by this factor, as it is controlled by a gene found on the non-homologous segment of X. So non-homologous segment of X, maybe it can be present in both males and females. But they told us that this abnormality, it affects only boys and not girls. Explain the absence of this abnormality in girls. Why do you think that this abnormality is absent in girls بس موجودة عند الميلز؟ عم بيقول لي this abnormality in hemophilia B, it affects only the boys, the males, and not the girls. عم بيقول لي explain the absence of this abnormality in girls. ليش ما بتبين بالجirls؟ First of all, لحتى تبين إذا الميل مين حيكون عنده X Y. So عنده 1X. However, female عندها 2X. So horn is a male, it appears in only one copy, since it has only one X chromosome. However, in the female, it should have two copies in order to be expressed if it's dominant or recessive, first of all. So here, explain the absence of this abnormality in the girls. Why? Maybe if it's present in two copies, this is lethal. Maybe it leads to the death of the female. This is the explanation of this abnormality. Maybe it's lethal and leads to the death of its homozygous carrier. So here it's lethal and it leads to the death of the females. That's why it's not expressed in girls. Document 1, it shows the pedigree of a family, one member of whom has the abnormality. So here document 1, عم بتفرجيني pedigree. Here we have normal man, normal woman, the hemophilic man, which is number 1. So this is number 1, it's hemophilic. Hemophilic meaning that it carries the allele of the disease. But they said show that this disease is recessive. They confirm it that the disease or the allele of the disease is recessive. How? This individual is male. It carries the allele of the disease, so it's XY. Okay? And this individual 3, it is XX. If the allele of the disease is dominant, then it should be propagated. Let's say it's dominant, let's give it D capital. So if it's XD capital Y, if the individual here is XD capital Y, so it should transmit XD capital to the female 3, who should be affected. But she's normal, so it's not the case. So the allele of disease is recessive in this case, since individual 1 is affected and it carries only one copy of chromosome X, so it's XD small y, and it's transmitted to the female 3. So this female 3, it should be heterozygous. So 100% it's heterozygous. 100% it should be XD. For example, let's say the allele of disease is D, so it should be XD and another allele XN, let's say. And here is XDY, the individual one. Okay, so till now everything is clear. Any questions before we continue? Everything is clear before we continue? Okay, so no questions. Great. So here we have xdy, and here we have xdxn. Why xdxn? Why it's not xdxd? Or why it's not xnxn? This female, it should carry the allele of the disease. It's normal. So it should be xnxn for 100%. Or it can be xnxd, for example. Maybe it's xnxd. But if it's xnxd, here we have possibility to have the individual 4 should be affected. So here it's xdy and here xdxn. So the female is here, it's heterozygous. They said in part 3, determine the genetic risk that the fetus will be hemophilic. If the fetus is going to be hemophilic, hemophilic meaning that it should be a hemophilic man. There is no chance to be hemophilic woman. Let's see why. Here, 3 it's XDXN. 4 is normal. So sure, it will be XN Y. So in order to be heterozygous, this is a half probability. So to be heterozygous, it can be XD Y. So it inherits the Y from the father 4 and the XD from the mother 3. So here to have this, its probability, it's half. So 1 over 2 to have this probability. Why? Because 1 over 2 to be a female, to be a male, sorry. So the probability to be a male is 1 over 2. And to give it the D. So it can be X and Y or it can be XDY. Or if the probability is to be female, the risk is going to be null. The risk is going to be 0. Why? Because any of the axes here, either xn or xd, but here in the male, it will inherit xn. So sure it will have xn. For 100%, it will have xn. And the other allele, it can be either xn, or it can be xn, xd where it's xn or xd, and in both cases it will be normal. So the risk for the fetus to be affected, if it's girl, it's null. So there is no possibility to be affected girl. While if the risk for the fetus to be a male, so the risk to be affected is 1 over 2. So going to make a quick revision about the 3 parts, 1, 2, and the 3, before I move to the other parts. So first, explain the absence of this abnormality in girls. They said it's located in the non-homologous region or segment of X. So it is a normal. This abnormality is absent in female. Why? Because it should be expressed in homozygous, and maybe it leads to the death of its homozygous carrier. Document 1 shows the pedigree of a family, one member of whom has the abnormality. They said show that disease is recessive. Why? Because individual 3 is normal, so it carries the allele XD from the father, sure, and another allele XM should be carried from the mother. That's why it's absent, it's masked, so it's recessive with respect to the normal dominant allele. Genetic risk for the fetus to be affected, so the genetic risk to be affected, if it's a female, the risk is 0, it's null. Why? If it's a male, the risk is 1 over 2. Moving now to the parts 4 and other parts. Let's move to part 4 now. Ultrasound scan was done to determine the sex of the fetus. It is revealed that is a boy. So it's a boy. So this individual, it's boy. So it has XY as sex chromosome in the normal case, since it's a boy. The boy having XY as sex chromosome in the normal case. I repeat, in the normal case. If it's abnormal, we have another issue. So here the doctor then prescribed the analysis of the DNA by the method of Southern blotting. They used the probe is used to determine or to distinguish between the mutated and the normal form of the gene. The obtained results they appear in document 2. They said specify the band that correspond to the defective allele. What the meaning of defective allele? Here they mean defective allele mean the allele causing for the abnormal gene or the gene that is abnormal or the mutated one. They said specify and they said justify the answer. So then no need to set the justify here. Already they said specify. So sure you have to justify. First of all, in order to specify the band that correspond to the defective allele, I have to look which individual is affected. Individual 1. Individual 1 is affected. Individual 1 is a male, so it has XY. How many alleles it will carry? Individual 1. Let's see. How many alleles for the normal or the affected? The normal or the affected, it will be expressed on the X. So it should carry only one allele. So the allele here, it showed that the probe, it has only one band of 1.3 kb, kilobase. So here it's 1.3. So 1.3 it's corresponding, so 1.3 band, kilobase band, correspond to the defective allele. Since individual 1, which is, has only one X, it shows this band. Individual 3, it's normal. It has two bands, 1.8 and 1.3. So it's heterozygous, as we said from the pedigree, and this confirms us. So it has two allele, individual 3. Individual 3 has XN, XD, if we give X symbols. Individual 4 is normal or is affected. Individual 4, first of all, it's a male or female, it's a male. So it carries only one band, so it's X and Y. Individual 7. What is individual 7? Individual 7, it's the fetus. They said that the individual 7, it's the male. They said that it's a male. So it revealed that this is a boy. But here, from the southern blotting, we have two bands. We have band of 1.8 kilobase and 1.3 kilobase. So here, what is the problem? Here it has two bands. So meaning that it has two axes. And since it's a male, so it has one Y. So here we have a special abnormality. So if you want to move to port 4, specify the band that corresponds to the defective allele. So the band that corresponds to the defective allele is the 1.3 kilobase. Why? Because the individual 1 is affected and it has only one copy. It has only one X chromosome. So it is the one that corresponds to the defective allele, which is 1.3. So here, the individual 1, it has XDY. There I use D and an, I use them from my own, okay? So I have to give symbols if I'm going to use that. Individual 3, it's normal, but it's heterozygous, if you want to say heterozygous. So it has the normal allele and the effective one. So the effective one is XN and X is small. Individual 4 is male and is normal, so it has X and Y. Individual 7 is the problem here. They said fact, identify from the DNA analysis the problem, the child that will be born. So here, what is the problem? It has two bands, one for the normal and one for the defective allele, and it's male. So he has two X chromosomes, X, X, and Y. So here the abnormality, it's what? The decline filter syndrome. This is the abnormality that the individual is suffering from. It has a Klinefelter syndrome. This person, if I want to ask a question, this person is sterile or fertile? Justify your answer. Let's see if you remember these. This individual is sterile or fertile? Yes. What do you think? This individual is sterile or fertile? Justify your answer. Yes. Great. 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 This individual has XXY, so he has abnormality in the sex chromosomes. So this abnormality in the sex chromosomes, so it will affect the sex chromosomes. So this means that this abnormality it affects the sex chromosome, so it's said to be sterile. However, if the abnormality is affecting the autosomes, it will be fertile, like Klinefelter syndrome. It's a sex chromosome abnormality, so it's affecting the gonosome. It's a gonosomal abnormality, so it's going to be sterile. The doctor completed the diagnosis by establishing the karyotype of fetus from document 3, except established based on documents 2 and 3 the diagnosis of the fetus. So the fetus said to be a male. The that is a male. So from this, we have XXY. So it's normal related to the hemophilia, but it has another abnormality, which is the Klinefelter syndrome. Specify the stage of meiosis at which the abnormality will take place during the anaphase, during the segregation or the separation of the gametes during the anaphase. Here, the allele of the hemophilia is lethal in the homozygous state. This explains the absence of this disease in the case of females. So the girl has two X chromosomes. She has, for example, they gave her XHXH, so she dies before birth. The disease is carried by the X chromosome. So individual one has only one X, which carries the allele responsible for the disease. So it's XH, so it will transmit it to the daughter. The daughter should be its normal girl 3, so it's recessive and it's masked. Fetus 7, it has heterozygous mother, which is 3. So if it's a boy, there is a risk of 1 over 2 to have the X chromosome carrying the hemophilia allele. However, if it's a girl, the risk is going to be null because there is a healthy father cannot give her a less healthy X or one normal X, X normal. The band that corresponds for the disease or for the defective allele of the hemophilia is 1.3 kb band because individual one who is sick has only one X, so it's only this band, so it's 1.3. The fetus is a boy, as we said in part 5, but he has only one X chromosome. Then he has only one band, according to this DNA analysis. But according to this document, it presents two bands. Therefore, it's a boy with two X chromosomes. So fetus 7 is non-hemophilic boy. It has no hemophilia. Since it has XXY, so it has the Klinefelter syndrome. What stage does this problem occur at what stage of the meiosis? So the meiosis during the anaphase. Why? Anaphase of equational or the reductional during the reduction and division, during the disjunction of the chromosomes, either XX or XY. Because upon the DNA analysis, we have two different bands. So we have two X's and not two chromatids of the same X chromosome. In this case, the father or the mother, they could be the origin during the segregation of these gametes. So either the male, the father, or the female, the mother, will be the origin of this disease. So this question is very important. So you have to get familiar for the absence of one of the diseases in case of X. Whenever it's absent in the female, this means that the allele of disease is lethal in the case of homozygous. It cannot be expressed in the homozygous state. So this explains what we are talking about, and this explains that the disease is dominant or recessive. Part 2. So here in part 2, show that the disease is recessive. Whenever you give the symbol of the disease recessive, so immediately you have to give symbols if you want to use the symbols like we did. I use, for example, M for the normal and D for the disease. or you can use N for the normal and H small for the disease. So this is related to the pedigree and related to that. Moving now to the DNA analysis of the thousand blood technique, we have the bands, so you have to read the bands and you have to pay attention to the question. It's found on the X. So whenever it's found on the X, this means that it is only one copy in the case of male and two copies in the case of female. But here the surprise was in the case of the fetus, we have two X's and it has another abnormality which is the Klein-Filter syndrome and it's normal. Why? Because it's carrying one band which corresponds to the normal and one band corresponding to the defective allele. So here, this is the explanation of question number 4 for our worksheet. Tomorrow we're going to continue with our question number